we're ready to hit Chapter 26, which deals with phylogeny and the tree of life. So we're going to investigate the different ways that we can make a tree of life and look at taxonomy and how the different groups of organisms interact with each other on our man-made scale. We're going to have a lot of definitions in this particular chapter, so make sure that you keep up with these. I'm highlighting some of these right now. You might want to start another sheet just of definitions so that you can build some flashcards or however you wish to do your study skills. So phylogeny is the evolutionary history of a species or a group of related species. So with phylogeny, you're looking at how these groups are related to one another, but you're also looking at the evolutionary history and that common ancestor that exists at some point in time. Systematics is a field of biology that classifies these organisms and helps to determine their evolutionary relationships. Now, keep in mind that this can be kind of hard because not everything's living anymore. So when you're looking at things in evolutionary time, sometimes you're making assumptions. So we have to use different sets of data, and systematicists will use fossil records. They'll look at molecular data, genetic data that infers evolutionary relationships. So we use that word infer because really it's a guess based upon the evidence that we have. So it's a best guess, and a lot of times computer programs are used to help make this best guess, and we'll talk about this a little bit later. Here we have a phylogenetic relationship showing a common ancestor here at the base of our tree. And as we look at the branches of our tree, we can see the kingdom Plantae here where we have the flower. We can see the kingdom Fungi, the kingdom Animalia. Now looking at this particular tree, the kingdom Plantae is on a branch all by itself. This upper branch shows another divergence right here, and this is going to split into two major diversifications, one leading to the kingdom Fungi, one leading to the kingdom Animalia. So the suggestion is fungus is more closely related to animals than they are to plants. And this line of thinking hasn't always been the case. In fact, when I took botany, the fungi were included in the botany class, and it's thought that the thought of the time was that fungi were in essence plants that were a little bit different. So we know from different types of data today that this is not the case. So let's start with our first concept, 26.1, that looks at how phylogenies show evolutionary relationships. So here we've got another definition. So we have taxonomy, and taxonomy is defined as the ordered division and naming of organisms. So ordered division and naming of organisms. Now, you have to keep in mind that taxonomy is completely a man-made field. Okay, so this is based on characteristics that man has created and characteristics that make it easy for us to look at all of that diversity that's out there. We know about binomial nomenclature already, so we mentioned back, I believe, in Chapter 1 about Linnaeus, Carolus Linnaeus, published a system of taxonomy that was based on resemblances. And those resemblances is what things look like. Okay, so you're looking at things based upon homology. Well, let me back up. Not homology, but morphology. So based upon the characteristics that a person can see. So the key feature of his system is something that we still use today, and that is that the two-part names for a species. And we also use that hierarchical classification. Now, some of the names have changed over the times, but this system has really stood up to the test of time. So that's binomial nomenclature. Let's take another look at another slide. So the fact that you've got a two-part scientific name, that's the binomial part. Nomenclature is referring to the name itself. We know that the first part is the genus. We've talked about all of this already. The second part is the specific epithet, and I've mentioned this before, that we typically call the species. Okay, we know that the first letter of the genus is capitalized, and this entire species name is going to be italicized. You do not capitalize the specific epithet of the binomial. So both parts taken together is what actually names the species. You can't use the specific epithet by itself, but you can use the first letter of the genus along with the specific epithet to describe a species as well. And that's very commonly done. And you guys do it all the time in terms of talking about E. coli. The E stands for Escherichia. That's the genus. Coli is the species or the specific epithet. We recognize E. coli as being that particular species. Now, Linnaeus introduced this system for naming species by providing increasingly broad categories. So this is his hierarchical classification. We're left with, and you do need to know this, domain, kingdom, phylum, class, order, family, genus, and species. We will be spending most of our time, hang on just a second, my color changed on me here. We want that, and we want this, okay. Domain, kingdom, phylum, genus, and species. We will talk a little bit about class when we get to the arthropods. And I think that's it. We won't spend a whole lot of time on class of our taxonomic groups in lecture and lab. So mostly domain, kingdom, phylum, genus, and species is what we will focus on, but you do need to know the entire classification scheme. So domain, kingdom, phylum, class, order, family, genus, and species. A taxonomic unit at any level of the hierarchy is called a taxon. So a kingdom would be a taxon. A phylum would be a taxon. A genus would be a taxon. The broader taxa are not comparable between lineages. So it says here, for example, an order of snails has less genetic diversity than an order of mammals. Okay, so you have to think of an order as being a subset of a class. Okay, so you're looking within the lineage itself, not necessarily between lineages as the taxons being exactly the same. This is a figure that's giving you a visual representation of the differences between the different taxonomic levels. So down here we have the domains. Okay, so domain eukarya, domain bacteria, domain archaea, and we're picking one of the the four that's shown here, and we're blowing it up, and this one happens to be the kingdom Animalia. We're picking one phylum of the kingdom Animalia, which is Chordata, one class, which is Mammalia, one order, which is Carnivora, one family, which is Phylogeny, one genus, Panthera, and one species, which is the Panthera pardus. Okay, so this shows where we go from a single species of an organism of a single species down to the very broad category of our domain. So how do we link together this classification that Linnaeus brought about with phylogeny that's looking at these evolutionary relationships? The way that we can look at these in a fairly easy way is to look at phylogenetic trees. So phylogenetic trees, you're looking at a schematic diagram, and it's very similar to the one that Darwin had put forward all those many years ago, showing the base of the tree, or the trunk of the tree, as being the common ancestor, and the branches of the tree being those organisms that diverge from that common ancestor. Here's an example of a phylogenetic tree, and we're using the same example that we used with the panther. So here we're starting with the order Carnivora and branching out from there. So we've got a branch that's going to the Panthera pardus, or the leopard. Okay, so here we go to the family Felidae, to the genus Panthera. Our other branch is going to branch yet again. So we're going to branch into the family Canidae and Mustelidae. Okay, these are going to go on to branch yet again into the Lutra and the Taxoidea, and Canidae is going to become Canis, which will branch a little bit later on into the Canis latrans and the Canis lupus, the coyote versus the gray wolf. Okay, so this gives you an idea of what a phylogenetic tree looks like, and you can have branches at different points where the taxonomic level diverged from the previous level. Linnaean classification and phylogeny can differ from each other. Okay, so one thing I want to impress upon you with this chapter, this is kind of a difficult chapter to cover. There's so many different ways to make evolutionary trees. There's so many different ways to make taxonomic trees. There's so many different ways to show relationships. Okay, so systematists have proposed something called the phylocode. Okay, we'll highlight that, the phylocode. And this recognizes only groups that contain a common ancestor and all of its descendants. So, and I put down here in parentheses, this is a phylogenetic tree. Now, there's different ways to make trees, but any time you have that common ancestor and you have all of the descendants, you're looking at a phylogenetic tree of some sort. So this phylogenetic tree represents, in essence, a guess, a hypothesis, if you will, about the evolutionary relationships that have occurred in the past. Now, you have to remember, we weren't around back then, so these evolutionary relationships are very much a guess. Okay, each branch point is going to represent a divergence of two species. So this would be where you had one species that is going to branch and become two species. Okay? Sister taxa are considered to be the groups that share an immediate common ancestor. Okay, so sister taxa are, for the most part, closely related to one another. A rooted tree includes a branch to represent the last common ancestor of all the taxa that are present on that tree. So a rooted tree, if you will, you think of that common ancestor as being the base of that tree, the trunk of the tree. So down there, that organism, that species that's holding the entire tree together, so to speak. A basal taxon is defined as a taxa that's diverged very early in the history of a group, and that basal taxa, basal refers to the base, so close to the base. So this is going to be close to the common ancestor. So this will originate near the common ancestor of the group. So that's a basal taxon. A polytomy or polytomy, This is a branch from which more than two groups emerge. Phylogeny is a branch from which more than two groups emerge, and we'll take a look at this too. We've got a nice diagram of this. Okay, so this is figure 26.5 in your book. Here we have that, in essence, that common ancestor. Okay, so this is the root of our tree. So this is our common ancestor, and we're going to diverge into two branches here. One branch is going to become taxon G. Now, taxon G is going to be considered the basal taxon because this particular species has remained unchanged from the time that it has diverged from that common ancestor. Okay? Up here, this divergence goes to a point, and at this point we have what we call a branch point, and this is where we get another divergence. So this particular species, whatever it was not around for very long. Okay, so it was present for a small period of time, evolutionary time, which can be hundreds of thousands of years, and then it branched into two other lineages, which is what we see here. One lineage, the one here at the bottom, is going to lead to that polygamy that I had mentioned before. So this leads to taxon D, taxon E, and taxon F. So these are three branches that came from this particular common ancestor that diverged equally at the same time. Okay, so as it says down here, an unresolved pattern of divergence. So usually when you see a branch point, you see it branching into two. In this particular case, you've got three species that are very similar, and yet we don't know exactly what happened to make them diverge. Okay, so the evidence isn't clear. So we're going to diverge up here. Taxon A is going to be up by itself, and this particular lineage is going to divide yet again to give us taxon B and taxon C. Now these are considered sister taxa because they're very close to each other in terms of divergence. Okay. If you wanted to look at taxon D, E, and F, those would be considered sister taxa as well. So what can we and what can we not learn from phylogenetic trees? So when you're looking at a phylogenetic tree, you're looking at patterns of descent. You're not looking at any sort of phenotypic similarity. Okay? Just patterns of descent. Phylogenetic trees do not indicate when the species evolved or how much change occurred in that lineage. Okay, so you're showing that these evolved basically early on or recent, but it's not suggesting any sort of measurable amount of time. It should not be assumed that a taxon evolved from the taxon next to it. And I want to back up just a second. So taxon G is not very closely related to D, E, and F. Okay? Taxon B and C is not closely related to D, E, and F. Okay, so you can't assume that just because it's right next to each other that a recent evolution has occurred between those taxons. So let's move on to the next concept here, 26.2. It says phylogenies are inferred from morphological and molecular data. Okay, so it goes on to say to infer or to make guesses about these phylogenies, systematists gather information about morphologies, how things look, about their genes, and about the biochemistry of living organisms. So we can work with living organisms. We can discover how these living organisms actually do look as they are alive, how their metabolism works, all of the interesting stuff we can see with the living organism. We can use what we call molecular and morphological homologies. So we've defined homologies already in class. You guys know that a homology is similarities due to shared ancestry. Okay, so when you look at morphological and molecular homologies, you're looking at specifically the phenotypic and genotypic similarities due to the shared ancestry. Okay, that's the homology. So phenotypic is what you can see. So what you can see is morphological. And genetic is going to be at the molecular level, looking at the DNA and looking at the proteins. So these homologies would be based upon similarities due to that shared ancestry. Now, if you do have similar morphology or similar DNA sequence, then organisms that share these similarities are going to be more closely related to one another than organisms that have different structures or different sequences. And that just makes sense, because the more differences that there are, then the more differences that have accumulated over time. But we have another word that we need to be aware of, and that word is analogy, or analogy. Analogy is similarity due to convergent evolution. Similarity that is due to convergent evolution. So homology, you're looking at shared ancestry. Okay? Analogy, you're looking at that similarity where two things come together. Okay, so you don't necessarily have that common shared ancestor. Yes, you're going to have a common shared ancestor because we all came from the same organism at some point in time. But once you go back to a certain point, then that shared common ancestor is kind of a moot point. So analogy is due to structures, morphological and even genetic evidence that have arisen separately and have shown up in organisms. So convergent evolution occurs when similar environmental pressures and natural selection is going to produce those similar adaptations in organisms from different evolutionary lineages. We looked at the flying squirrels a little bit earlier on in the semester, and that would be a convergent evolution. So you get the same feature. It's an analogous structure. They arose separately from each other, but again, very similar environments led to the formation of that structure. Okay, so we've gone over that bat and bird wings are homologous as forelimbs, but analogous as functional wings. So the fact that you've got bat and bird wings, the wing part is different. So bats can still use their forelimbs. Birds cannot. So birds have completely given up their forelimbs in terms of flight. But if you watch a bat, they can actually still use their forelimbs functionally and also for flight. So here you've got homologous structures, but they're still analogous structures when you look at them in a different way, so to speak. It goes on to say analogous structures or molecular sequences that evolved independently are also called homoplasies. Okay, so let me highlight that one, homoplasies. So you should be aware of that word because you'll see it on and off again and again. So how do we distinguish homology from analogy? You go back to the fossil record and you look to see how something arose, and you look at the degree of complexity that has arisen with that structure. So the rule of thumb is the more complex two similar structures are, then the more likely it is that they are homologous. Okay? The less complex they are, then they have probably arisen through two different pathways. So that's a general rule of thumb. Evaluating molecular homologies. This is done with the computer. So it says systematists use computer programs and mathematical tools when analyzing comparable DNA sequences from different organisms. The DNA sequence of a gene, and I think I've shown you this in lab, is very long. So it can be hundreds to even thousands of base pairs long. And this is a little bit too difficult to look at just by yourself. So there are computer programs that are housed at the National Center for Biotechnology Information, which is funded by the federal government, that stores DNA segments, and you can run a program to compare a segment that you have with a segment that's in the database. So it makes analyzing quite a bit easier. This is an example of looking at similarities or how things could have changed over time. We're looking at two sequences of DNA. And 1 and 2, pictured here at number 1, they're exactly the same. As we go through evolutionary time, sequence 1 has a deletion. Sequence 2 gains an insertion of 3 nucleotides. By the time we look at organism 3, we've got a place where our deletion has occurred, and we have a place here where our insertion has occurred. Now our DNA sequence for number 2 has become quite a bit longer, and our sequences are now quite a bit different when we line them up next to each other. So looking at number 4 down here, if we place in lines symbolizing an event that could have happened, then we can see how these similarities may be there, or in evolutionary time, how they could be the same. And computer programs can pick these up. Okay, so they can do those insertion of the lines for you very easily and do the comparisons that way. As you're looking at evolutionary data, it's very important to distinguish homology from analogy. And we use mathematical tools, we use computer programs to help identify these homoplasies or coincidence, if you will. There's a field called molecular systematics, so let's highlight that one, molecular systematics, that uses DNA and molecular data, so that would be our protein sequence, to determine evolutionary relationships. And again, you're looking at using computer programs to help you do that. Okay, so this is how we could pull out similarities based on these two sequences. So everything here in yellow is showing you things that are the same. If you didn't have a computer program, it might be difficult for you to pull out these similarities. So could this particular structure be homologous? It's very well possible that it could be, but not a lot of similarities between the two. Okay, concept 26.3, shared characters are used to construct those phylogenetic trees. So once those homologous characters have been identified, then you can infer a phylogeny. So remember, you can only use homologous characteristics. Analogous characteristics can't be used because there's no common ancestor. Okay, so we're only using the homologous characters. Cladistics. Okay, so let's get cladistics down here. So we've got some definitions. Cladistics is a type of phylogenetic relationship that groups these organisms by common descent. Okay, definition of a clade is a group of species that includes an ancestral species and all its descendants. Then it goes on to say that clades can be nested in larger clades, but not all groupings of organisms qualify as clades. Okay, so I mentioned this in lab, that a clade is a taxonomic level, and that taxonomic level has a common ancestor. Okay, and the next taxonomic level could also be a clade. Okay, so you've got to kind of keep that in mind. Let's take a look at our next slide here. A valid clade is monophyletic. And monophyletic, mono means one. Phyletic, you're looking at an evolutionary relationship. So this means that it consists of the ancestor species and all of its descendants. So you've got the common ancestor and every organism that's come from that common ancestor. Monophyletic, back to that one common ancestor. Paraphyletic is a grouping that consists of an ancestral species, but just some of its descendants. Okay, so you're not getting all of the descendants in the particular grouping, so it's not considered to be a valid clade if it's paraphyletic. Polyphyletic, poly means many, so here you've got a grouping that has different ancestors. Okay, so while you can have those groupings that are represented, it's not considered to be a valid clade in terms of cladistics. Okay, so let's take a look at this. We've got this figure, this is in your book, and here's our monophyletic group. Okay, so looking at group 1, this is a clade. Here is our common ancestor. Okay, so that's going to branch. We've got a branch that goes to A. We have another branch that's going to branch again to give us B and C. This is a valid, group 1 is a valid clade because this is considered to be the common ancestor for that group. Okay. Group 2 is a paraphyletic group. Here is the common ancestor. Okay, so we're going to branch and then branch again into D and E. The other way, the other branch point is going to branch into F, but there's another branch point here that's not included in this one's G. Okay, G is not part of group 2. It's considered to be a paraphyletic group. If it included G, whatever that lineage is, then group 2 would be considered monophyletic as well. But it's not including the lineage G. Okay, that's why it's considered to be paraphyletic. Polyphyletic, if you look here at group 3, we're not seeing a common ancestor. Okay, so you're looking at A, B, C, and D, but we're not looking at the common ancestors of these groupings. If we did, we would have to look at this one here. Okay, so the polyphyletic group here is not considered a clade, although it can be considered a valid grouping. It's not considered a clade. Shared ancestral and shared derived characteristics. So in comparison with its ancestor, that organism has both shared and different characteristics, and this kind of makes sense. So if you go back to that common ancestor, you've got a set of characteristics that makes up that organism. Okay? Now, over evolutionary time, there's going to be accumulations of differences that occur within that species of organisms, and sometimes that accumulation of changes is enough to make another species. Okay, so that's when we get a branch point. Now, that new species is going to have those ancestral characteristics, but they're also going to have newer characteristics that have evolved since the branch point. or even multiple branch points, if you will. Okay? A shared ancestral character is a character that originated in the ancestor of the taxon. So that would be the characters that the original species had. A shared derived character is going to be a characteristic that has evolved since you've had branch points. Okay, so this would be in the new species, it's going to be a characteristic that that common ancestor didn't have. So it says here, an evolutionary novelty unique to a particular clade. So that's going to be that new characteristic that made that species into that new branch point. A character can be both ancestral and derived depending on the context. So I left that in there just because I want you guys to be aware of that. It doesn't come up that often. Okay, so know the definition of the shared ancestral character and the definition of the shared derived character. We can infer phylogenies using these derived characters. So when you look at these evolutionary relationships, it's very useful to know in which clade that shared derived character first appeared. So when then did that new characteristic first come about that you see in the later times? So let's take a look at this. We've got a characteristics table here in A, looking at different taxa. So the character table includes a vertebral column, hinged jaws, 4 walking legs, presence of an amnion, and presence of hair. And we look at several different taxa going from the lancelet to the lamprey, to the bass, to the frog, to the turtle, to the leopard. So we're going to look at these characteristics and when they came about. So here's our common ancestor right here, and the outgroup is the lancelet. Okay, so this is that basal taxon. So this is that taxa that is close to that common ancestor, if you will. Here we have when the character of a vertebral column originated. So this was way back in evolutionary time. Everything that comes from after, well, let me back up. Everything that comes after this branch point is going to have a vertebral column. So everything from the lamprey on down to the leopard is going to have that vertebral column. Here we have hinged jaws. Okay, the lamprey does not have hinged jaws because the hinged jaw was derived after the vertebral column. Okay, so the hinged jaw is more recent than the vertebral column is. Okay, so everything from the bass fish all the way down to the leopard is going to have a hinged jaw. Next we evolve the 4 walking legs. The frog to the turtle will have the 4 walking legs. Amnion includes the turtle and the leopard is the only one with the derived characteristic of hair. Okay, so that's how you read this particular phylogenetic tree, and you look in respect in evolutionary time as to when these characteristics came about. Okay, so we placed upon this phylogenetic tree how these organisms would be related to one another in terms of accumulation of these derived characters. So the leopard has all of them. Okay, and we can see that here. Here's the leopard, contains all of the derived characters. Okay, the outgroup is a species or a group of species that's closely related to the in-group, which is the species that are being studied. So our outgroup in this case was our basal species. So it diverged before that in-group came about. So we include the out-group to have something to compare to, okay? So that's something that does not have the shared derived characteristics, but it does have the shared ancestral characteristics that all of the groups share. Okay, so characters shared by the out-group and the in-group are ancestral characters, and I'd mentioned that already, so you've got that in writing. Now, there's different ways to make these phylogenetic trees. So you can have phylogenetic trees that have the branch lengths that are exactly the same. So in some trees, that length of the branch can reflect the number of genetic changes that have taken place in a particular DNA sequence within that lineage. Okay, the length is showing you the differences, if you will. Let's back up just a little bit. The length of the branch can reflect the number of genetic changes that have taken place in a particular DNA sequence. Okay, so here we've got the fruit fly. There's our lancelet again, a zebrafish instead of the bass, a frog, a chicken, a human, and a mouse. Okay, by looking at the lengths of these lines, the human and the mouse share a lot of similarities in terms of their DNA sequence. Okay, chicken is a little bit closer to the human and the mouse, the frog a little bit more removed, and so on and so forth. Okay, so when you compare the mouse to the fruit fly, there's not a whole lot of similarity between the two based upon the length of that line. Okay, the shorter the line, the more the same they are. You can have other trees, other phylogenetic trees where the branch length can represent chronological time. Those branch points are typically determined from the fossil record. Okay, so this one, we go from the millions of years ago to the present, and you're looking at kind of the same idea here. So Drosophila, the lancelet, zebrafish, frog, chicken, human, and mouse, and you can see that the human and mouse diverged together in fairly recent times. Chickens go back to the Paleozoic, and the lancelet and your Drosophila go all the way back to before 542 million years ago. Pretty amazing. There's something that is called maximum parsimony and maximum likelihood. Maximum parsimony and maximum likelihood. So systematics is difficult because you're trying to guess how things are related to one another. Okay, so keep in mind that we weren't around back in evolutionary time. So if we weren't around, then we have to make guesses. Okay, so maximum parsimony assumes, okay, here's maximum parsimony. It assumes that the tree requires the best tree requires the fewest evolutionary events. Okay, so the fewest appearances of shared derived characters is the most likely path that the organism took to evolve. Okay, so maximum parsimony is an accumulation of the least amount of derived characteristics. Okay, maximum likelihood is, in essence, how likely this is to occur. Okay, so based upon what we know about DNA and about how DNA changes over time, maximum likelihood is the percentage, basically, that this is going to happen. Okay, mathematical formulas. So both maximum parsimony and maximum likelihood can be, let me go forward a little bit, can use computer programs to show trees that fit both maximum parsimony and maximum likelihood. Okay, very hard to do by hand. Too much information. So this was derived from the percentages shown between DNA sequences. Using that information, which tree is more likely to be true. Okay, so we looked at this tree before, and here's the data for us, 30%, 40%. And here's our flower, our fungus, and our animal. And here's our percentages here. So 20% change, 5% change accumulated here, 15% for both of these. Okay, so tree number 1 was considered to be most likely to be correct. Here's another tree. Here we have our plant, our animal, and our fungus. Here is a representation of 10% change, 15% change, and 25% change, with the fungus only accumulating 5%. And based upon data, the percent differences between the species, tree number 1 fits that data much more better than tree number 2. Okay, so maximum parsimony would be the data that we have between the differences between species, so how much derived characters can accumulate, and maximum likelihood is coming up with those percentages and which one's more likely to occur. Phylogenetic trees as hypotheses. So the best guess, the best fit for these phylogenetic trees are the ones that fit the most data. So the more morphological, the more molecular, the more fossil data that can go into building these trees, then the truer the trees actually are. So there's something that's called phylogenetic bracketing that allows us to predict the features of an ancestor from the features of its descendants. And this phylogenetic bracketing is kind of, well, we know that the extant species looks like this. Extant means living. So then the extinct species may have looked like this based upon characters and organisms that we know. So the example that they give here is that phylogenetic bracketing allowed us to infer a lot of the characteristics of dinosaurs. And if you ever follow any of this data, this information changes from time to time. So sometimes they back up and say, yeah, this dinosaur that we thought was scaly actually had feathers. So once we come across data, sometimes we change our minds, which is the beauty of looking at phylogenetic relationships. So here's an idea of looking at those relationships. Birds are very closely related to dinosaurs, and we can look at the characteristics of birds to kind of determine how these dinosaurs might have looked, especially when we consider crocodiles, lizards, and snakes, since our common ancestor is back here. Okay, so you would look at all of the characteristics of both the out group and the in group, so to speak. Okay, moving on to the next concept. An organism's evolutionary history is documented in its genome. So you can compare nucleic acids or other molecules for that matter. You can look at amino acids of proteins as well. And you can chart those changes that have occurred over time. don't spend a lot of time with this, but DNA that codes for ribosomal RNA changes very, very slowly. So you can look at the DNA of ribosomal RNA and see when changes occur. Mitochondrial DNA, so the DNA that's present in mitochondria, evolves very rapidly. So you can look at recent evolutionary events when you look at mitochondrial DNA. Gene duplications and gene families can lead to increasing the number of genes in a family. So a lot of times when you get gene duplication events, it's easy to trace back to a common ancestor. So it's easy to trace when this gene duplication actually did occur. Orthologous genes, this is something that you should know. Orthologous genes are found in a single copy in the genome, and they're homologous between species. So orthologous genes are species specific. So they can diverge only after speciation occurs. Okay, so orthologous genes can only change after speciation occurs. So here's the formation of orthologous genes. So here's our ancestral genes, and here we have species A and species B. So we've got an insertion of red and an insertion of yellow in these particular sequences. So these have evolved since we had this ancestral species. Okay, so this is a product of speciation. Paralogous genes come from gene duplication, paralogous genes. Okay, there we go. These results from gene duplication. So you can have these in more than one copy within the genome. So these often diverge within the clade where you see them, and the more copies that you accumulate, you very oftentimes get new functions along with that. And this shows the formation of a paralogous gene within a species. Okay, so there's our ancestral gene. Here we get the formation of a duplication, and then divergence can occur possibly by an insertion after that gene duplication event. Orthologous genes are widespread, and they extend across widely varied species. So humans and mice, and we saw that example a little bit earlier, diverged about 65 million years ago, but 99% of our genes are orthologous. Okay, so 99% of our genes are orthologous. Gene number and complexity of an organism aren't strongly linked. Genes perform many functions. Genes can make more than one protein within cells and within organisms. So just looking at how many genes are present within a particular species very oftentimes has no meaning whatsoever. Okay, so don't write anything down from the slide. Just kind of, keep in mind that the size of the genome doesn't necessarily tie in with how complex the organism is. So the last concept here, new information continues to revise our understanding of the tree of life. So molecular systematics, so looking at the sequence of DNA, looking at the sequence of amino acids within proteins, this is our molecular systematics, and this has completely changed how we classify organisms. It changes. It's man-made, and it changes, and I've mentioned that before. We went from 2 kingdoms to 3 domains. So when I was going through college, I learned 5 kingdoms: Monera, Protista, Plantae, Fungi, and Animalia. So Monera was the prokaryotes. Protista, as we've recently learned, has been broken up into many different groupings now. The plants, the fungi, and the animals. Now we've got the 3 domain system. So we've made a classification that's above the kingdom level to include the bacteria, the archaea, the eukarya. Our three-domain system actually comes from the sequenced genomes. I don't know how this classification scheme got in there. So, wow, things change. It's thought that this leads to our branching of our three domains. So it's thought that our common ancestor, or the trunk of our tree, if you will, is right here. So bacteria is kind of on a branch by itself, and then our other branch diverged to give us the eukarya and the archaea. So those ancient bacteria are actually closer to eukaryotes than they are in terms of relationship to bacteria. And this is what I just mentioned to you guys. So the tree of life that we just saw is based pretty much on ribosomal RNA genes, because these guys have evolved the slowest over time. We've got issues because there's been a lot of interchanging of genes between organisms in different domains. We call this interchanging horizontal gene transfer. So this is the movement of genes from one genome to another. And this horizontal gene transfer, and we'll talk about this a little bit more with prokaryotes, exchange of transposable elements and plasmids. So in essence, what this is small pieces of DNA that can be exchanged from one organism to the next, from one cell to the next. Viral infections can lead to horizontal gene transfer. So viruses can bring strands of DNA with it from other organisms. And fusions of organisms can lead to horizontal gene transfer. So making this tree of life can be very difficult because of this horizontal gene transfer, okay, because of information being passed between species, if you will, that wouldn't normally pass information. So the question here is the tree of life really a ring? So it's thought that eukaryotes may have arisen as a fusion between a bacterium and an archaean. So it's thought that a ring may be a better representation of how our life came about, or who our common ancestor may be. So this would be the branch point of our bacteria, the branch point of the archaea, and then basically this single divergence of our eukarya from there. So this has been suggested. Has it been readily accepted yet? No, we're not there yet. So we'll see what comes about here in the next couple of years. This has been kind of a difficult chapter. There's a lot of concepts here that you may not have ever covered before. This is kind of an introduction of going into taxonomy. So the next chapter we're going to pick up and talk about our prokaryotes, so the domain archaea and the domain bacteria. So make sure that you understand this chapter, because it's very important in knowing how to place all of these relationships that we talked about, how to put things in order in terms of evolution. Okay, thank you so much. I hope you've enjoyed chapter 26.